In this chapter, we're going to learn about the defense system. Immune system in vertebrate provides a defense mechanism against foreign parasites such as viruses and bacteria. Three major properties are specific recognition of foreign molecules, ability to destroy the foreign parasites, memory that allows a more rapid response to the second infection. Specifically, three molecules relevant to immune systems are antibody, especially immunoglobulin G, produced by B cell, and T cell receptor, provided by T cell to destroy the infected cells. All cells have basically the MHC molecule on the surface, major histocompatibility complex. You can see here some two basic immune system. First in A, the humoral, the defense mechanism, binding of antigen to the B cell triggers a process that eventually activates the B cell to synthesize and secrete many copies of antibody. In this process, a small amount of antigen can elicit an amplified and specific immune response that helps to clear the body of the source of the antigen. The antibody-tagged pathogens are recognized and destroyed by macrophage and other effector cells in the immune system. The second one is cell-based immune system. Binding of a T cell receptor to an MHC antigen complex triggers activation of a T cell to kill infected cell or elicit help from other cells in generating an immune response against phagocytosed material. There are two classes of MHC, MHC1 and MHC2. Most cells, except central spinal cord, has MHC1. MHC molecules binds degraded fragment of peptide antigens generated inside infected cells or taken up by specialized phagocytotic cells and display the fragment at the cell surface for recognition by T cell. The MHC1 antigen complex can be recognized by the CD8 plus T cells and killed. The macrophage taking over antigen will make MHC2 antigen complex. In this case, this complex can be recognized by CD4 plus T cells and causing the immune defense, including activation of a B cell, something like that. The basic structure of all immunoglobulin molecules comprised two identical light chain and two identical heavy chain linked together by disulfide bond. There are two different classes or isotype of light chains, lambda and kappa. Heavy chains, by contrast, have 5 different isotypes that divide the immunoglobulins into 5 functional classes: immunoglobulin G, M, A, T, and E, shown in B. We'll discuss immunoglobulin G class in this chapter, which is measured in normal human serum. Immunoglobulin G is built up from 2 copies, each of 2 different polypeptide chains: heavy chain in red and light chain in blue. The light chain folds into 2 domains: VL, with a variable sequence between different immunoglobulin G molecules, and CL, with constant sequence. The heavy chain folds into 4 domains: 1 variable VH, and 3 constant domains CH1, CH2, and CH3. The subpart bridges connect 4 chains, here, here, and here. The antigen binding sites are at the end of the variable domains, here and here. Each domain has 110 residue lengths. Certain regions within variable domains for light chain and heavy chains show high degree of sequence variation. These regions determine the antigen specificity and are called hypervariable region or complementarity determining regions, CDR for short. There are 3 regions: CDR 1, 2, and 3 in each variable domain. The sequence of large number of variable domains in light chain have been compared, and the sequence variability is plotted as a function of residue number along the polypeptide chain in x-axis. The 3 large peaks in this diagram, in dark color, correspond to the 3 hypervariable regions in A. These hypervariable regions vary both in size and in sequence among different immunoglobulins. However, all variable domains and constant domains each share sequence homology, suggesting the common ancestor for each domain. Antibody diversity is generated by several different mechanisms. Human body generates as many as 50 million new antibody producing B cells every day. For the heavy chain gene pool, variable domain is encoded by 3 types of segments. Approximately 1,000 different V, which encode about 90 the initial the residues in heavy chain variable domain, about 10 different deep segments which encode the CDR3 but varied in their length, and 4 different J segments encoding the rest, the 15 residues. The DNA for the variable domain is assembled by random joining of one of each these three segments into a single continuous axon called combinatorial joining that occur in about 40,000 different ways. The joining of these segments is not precise at the junction of D with V and J, and this creates additional junctional diversity. Extra nucleotide can be added during this joining procedure, giving still further diversity. In case of light chain, the similar process occur, except this has no this segment. Altogether, 90,000 different heavy chains and 30,000 different light chains can be made. So for the complex of these two components, you can get 270 million different types. Furthermore, the actual number of antibody will be several orders of magnitude higher than this number, because point mutations can be introduced into the exons for the variable domains by somatic hypermutation. Here, V-D-J joining process involved in making a heavy chain in the mouse. During B cell development, the chosen V gene segment, in this case V3, is moved precisely next to the chosen D and J segment, D2 and J2 in this case. The extra J genes, J3 and 4, and the intron sequence are transcribed along with the V3, V2, J2, and C segments, and then removed by RNA splicing to generate messenger RNA for this specific heavy chain shown here. The complete immunoglobulin G molecules have been difficult to crystallize because of the conformation of flexibility in the hinge region between the CH1 and CH2 of the heavy chain. Immunoglobulin could be partially digested with the proteolytic enzyme papain into two identical FAB and one FC, which their names coming from this kind of flame: FAB, F fragment antigen binding, FC from fragment crystallizing easily. Three groups in 1970s determined the initially different the state of the immunoglobulin. It turns out all domains shows the similar structure called the immunoglobulin fold. Basically, immunoglobulin fold is a Greek barrel. Here you can see the constant domain, where you can see the 7 anti-parallel betas with 2 layers, one with 3 beta and the other with 4 beta. In the blue color, topology at the bottom shows the Greek barrel topology.